To better understand the connections between billions of neurons, the complex interactions of different brain areas, and what goes wrong to result in neurological disorders so that we can develop new treatments, there's no way out of it. Samples and data are needed. And to get these samples and data, of course, patients are needed. Again and again, at the risk of repeating ourselves, they are essential for the advancement of science. Essential for the development of resources like the Neuro's CBIG repository. Big for big, yes, we could say it's a big resource, but also CBIG, B-I-G, for Clinical Biologic Imaging and Genetic Repository. Please welcome the director of the CBIG repository, Jason Karamchandi.
Good morning, everyone. Thank you for joining me here today. My name is Jason Karamchandi, and I'm a neuropathologist here at the Neuro. I'm an associate professor in the Department of Pathology and the director of the Neuro's Open Science CBIG repository. So I'm showing you a picture here of a peripheral blood smear from a young boy. This image was provided to me from one of my colleagues from residency. And unfortunately, this is not a normal blood smear. There's all these purple blobs that don't belong there. And this is actually a smear from a young patient with leukemia. Now, leukemia was identified in the late 1800s. Less than 100 years after the identification of the disease, the first effective therapy was deployed in humans in the 1950s. Since then, not a single decade has passed in which we haven't made progress at the treatment of this disease. If you look at the curve on the left of your screen, you'll see that the survival rates for patients with leukemia have continuously improved over the last 7 decades. In fact, this blood smear from this real patient was successfully treated using modern chemotherapies and treatment regimens. So a disease that killed everyone 100 years ago now has greater than a 90% 5-year survival in 2020. Let's compare that with Parkinson's disease. Dr. Parkinson's actually characterized this disease in 1817. More than 100 years later, Dr. Carlson discovered that the administration of L-DOPA in animal models reduced the symptoms of Parkinson's disease. 10 years later, that was tried in human beings successfully. So now 60 years, it's been 60 years since we've really substantially changed our treatment of Parkinson's disease. I'm being a little bit cheeky because we have added on MAO inhibitors and other small tweaks, but we've really made no substantial progress. And why is that? What's the difference between leukemia and Parkinson's disease? One of the difference is the access to the human tissues that drive the disease. Those leukemic cells I showed you in that first image are extremely easily obtainable with a simple blood draw. We are unfortunately not able to harvest the midbrain dopaminergic neurons from patients with Parkinson's disease, though. So we're hindered because we don't have access to the tissues that allow us to understand these diseases. So we feel a desperate need. All of us are impacted by neurological diseases. When I give this talk in person, I ask anyone who's affected by neurological disease or has a loved one that's affected with neurological disease, please raise their hand. And reliably, every hand in the room goes up. In fact, neurological disease is expected to exceed cancer as the second leading cause of death in Canada within 20 years. So we have this sense of frustration and urgency, both as treating clinicians and as patients and members of these communities. One of the downsides of pandemic is that you're not able to join me here today in this historic institute. But this building has a near 100-year history of allowing patients to come here for excellent clinical care, where they simultaneously participate in research. A patient walking up these steps in 1938 to get incredibly high-end neurosurgical intervention was also participating in mapping out the human brain. So with these challenges, under the leadership of Dr. Guy Rouleau, the director of the Institute, the Neuro adopted an open science philosophy. This was not a cavalier decision and actually followed 1.5 years of stakeholder consultation, where I think most importantly, we came up with a definition of what open science meant to researchers and clinicians at the Neuro. And yesterday in one of our brain dates, we started talking about the definition of open science. And I think it's worth considering that that term means different things to different communities. The people who work in astroscience and the people who work in software have been doing this for decades. And we here in the health sciences and life sciences are really just learning about this and starting our exploration. So Dr. Rouleau established the guiding principles of open science in our building. All of you are here today because you have some interest in open science, and so I don't need to convince you about the positive aspects of open science, but it's really driven by a desperate need for effective therapies. This was really empowered by a substantial and transformative gift from Larry Tannenbaum that allowed for the creation of the Tannenbaum Open Science Institute. I'm going to quickly go over the 5 guiding principles because I believe that CBIG is a reflection of these principles that emerged from this long stakeholder consultation here at the Neuro. The first is, of course, the public release of scientific data. And this data must adhere to the FAIR principles, meaning that it's findable, accessible, interoperable, and reusable. All of our research partnerships, both industry and academic, are performed in an open capacity. And we're going to touch back on that a little bit later. Indeed, the creation of the CBIG repository was one of the founding principles of open science here at the Neuro. The Neuro doesn't support the development of intellectual property for basic science. That is, this is not anti-IP, it's about the when, not if IP is a good thing or a bad thing. It's just that producing IP early on in the scientific exploration doesn't help patients in the end. And finally, Dr. Rouleau is not walking around the hallway with a stick. We recognize the autonomy of investigators, scientists, and clinicians here at the Neuro, and this is a buy-in system. The main goal of the CBIG repository is to empower patients. We have very little to offer patients with some of these rare neurological disease, and by contributing biospecimens and data, these patients are directly helping scientific and industry collaborators to help advance our understanding of their diseases. So we are allowed to consent and collect materials from a wide range of patients, meaning both adult and pediatric patients with neurological diseases, as well as healthy controls. And of course, what we collect will depend on the nature of the patient's disease, and everything we collect is done with the appropriate clinical standards. So as I said, we collect a wide array of biospecimens, including blood, cerebrospinal fluid, brain tissue, skeletal muscle, saliva, skin, urine, and peripheral nerve. Many of these tissues allow us to produce cell lines, and we're going to explore that in a little bit. So blood, skin fibroblasts, and urine can all produce cell lines which can be used to do molecular interrogation of neurological diseases. In terms of imaging, we can collect both clinical imaging, meaning the scans and studies that are done as part of clinical care, but also research-specific imaging. And the Neuro is lucky to have one of the largest brain imaging centers in the world, certainly the largest in Canada, and that allows us to do imaging series that we would not be able to do in the clinical setting. And finally, we collect a wide array of clinical data. Importantly, we collect longitudinal clinical information. We see these patients multiple times over multiple visits, and as all of us know, neurological diseases have a way of waxing and waning in a way that we do think is important, particularly if you combine that information with therapeutic interventions. Most of our patients undergo some form of generic interrogation, and we'll talk about that a little bit more when we talk about our new database, but many of our patients undergo some form of whole genome interrogation. Importantly, and somewhat unique here in Quebec, is that every patient in this province has an electronic medical record. And while this dossier is not fully complete, it does contain a lot of important information, including which medications a patient is on, some basic lab values, and perhaps most importantly, all clinical imaging is centralized in this provincial database. We also collect disease-specific information in the form of customized patient intake forms and disease-specific questionnaires. And this is where it gets really exciting. This may sound like science fiction, but what you're looking at are two organoids derived from patient cell lines. So we can take blood from patients with neurological disease and perhaps even a genetically caused neurological disease. We can then reprogram those cells into stem cells and grow them in 3D cultures into organoids. We affectionately refer to them as mini-brains. And you'll see here these two organoids appear slightly different. The one on the right is pigmented, and that's because the medium in which it was grown allowed the neurons to differentiate into dopaminergic neurons that produce neuromelanin, which is why it's slightly browner than the organoid on the left. Now we have the ability to work with the cells that are involved in the pathology of human neurological disease. This is new. This is less than 10 years old. And we can also leverage another fairly recent discovery in CRISPR so that we can repair the genetic alterations that are implicated in the human neurological disease and create an isogenetic cell line. So we have a disease cell line and a control cell line. This is what this looks like under a microscope. I'm a neuropathologist, so I find this very exciting, but you'll see some of the cells here that are indicated by arrows are really mature-looking neurons or ganglion cells. And the slightly more purple areas are more evocative of the developing brain. So what we see here under the microscope is that these cells are really recapitulating what they do in human development. So now we're cooking with oil. Now, for the first time, we can take patient materials. We can create stem cell lines, repair those genetic alterations, and this work is done with the Experimental Drug Discovery Unit here in the Neuro under the direction of Dr. Fawn and Dr. Durkin, and we can produce these organoids. And this is a new biological reagent to explore human neurological diseases. And our hope is that this empowers new drug discovery, which will eventually be returned to the patient communities. So this is my fantasy graph here. 1950, we had our first intervention in Parkinson's disease. Over the next 10 years, I hope we develop a comprehensive array of patient-derived cell lines. We already have several dozen here at the Neuro and in CBIG. And with these cell lines, I hope we're able to develop targeted therapies that meaningfully impact Parkinson's disease within the next 10 to 15 years. So where are we now? CBIG launched in 2016, and we've now collected more than 30,000 samples from more than 2,700 patients. And I put here our recruitment rate, and I think it's worth addressing that our patients are incredibly motivated to participate in this project. In fact, I think we've had only 12 patients decline participation in the last 4 years. We've collected more than 1,000 patients with Parkinson's disease. Many of these patients, more than 80 of them, have undergone research-specific imaging here at the Brain Imaging Center. More than 800 of these patients have had genome-wide interrogation, either in the form of wide panel testing or high-depth single nucleotide polymorphism chips. Either way, it's a way of really examining the genomic backgrounds of these patients. We have more than 250 patients with amyotrophic lateral sclerosis, and again, almost all of these patients have been genotyped. We also have a growing collection of patients with neuromuscular disease. And Muscle biopsies are a fairly uncommon procedure, and we do a lot of them here at the Neuro. So we have a growing collection of human muscle tissue, which has been very carefully handled and snap frozen. And most of these muscle biopsies have paired blood draws to allow us to study neuromuscular disease. We also have a growing collection of patients with multiple sclerosis, and we're expanding into other areas, including autism and intellectual disability, frontotemporal lobar dementia. And an interesting facet of CBIG is that it was designed to aggregate extant collections of human data and biospecimens, meaning that other collections that were project-specific can be decanted into CBIG for secondary reuse. We have already participated in more than 20 collaborations, both academic and industry, and we partnered and collaborated with groups based here in Canada, Japan, the United States, and the United Kingdom. Now, we're going open today, but we already had 6 collaborations in our first 3 years, but since June 2020, there's a typo on this slide, we actually had 13 requests in the last couple of months. So this is starting to build momentum and pick up speed. I want to focus on the external research partnerships, which is one of the founding principles of TOSI, because our collaborations, even with industry, are conducted in an open capacity. And what that means is if we provide biological materials for an experiment to a company, the experimental results are returned to CBIG. The goal there is to avoid duplication and to allow other research groups to leverage that existing work. I have to admit I was a little bit skeptical that this would work with industry, but surprisingly, our industry collaborators have been very open to the open science philosophy. And I'm going to show you 2 examples here. Here's a collaboration with Merck in which we provided serum samples from patients with Parkinson's disease, and they looked for autoantibodies against alpha-synuclein. In a different collaboration with a group in Japan with Takeda, we provided peripheral blood mononuclear cells. And one of their assays was to look to see if we were providing them with high quality material. And even though these samples were shipped halfway around the globe, they had a viability which exceeded their expectations. This reflects our good laboratory practices for specimen collection and sample processing. This required a really unique and robust ethics and governance structure. So the framework that houses CBIG has undergone 2 substantial revisions, and it's built with open science at the core. We have a novel patient-centered consent form in which every patient receives a unique consent form depending on the nature of their disease or if they're a healthy control. We had to create a new open transfer agreement, which is unique at McGill, in which the legal documents that allow us to share data and materials is stripped of all the verbiage that would restrict other people to innovate and to create IP on their side. We've created a new form of consent codes to allow us to harmonize collections across institutions, and that's being published right now. Anyone who logs on today to the CBIG portal will see a community standards. It's the equivalent of a terms of reference that all of us are so familiar with when we download an app on our phone, and we agree to certain terms and conditions. And really in this document, it's about agreeing to make sure we're doing what's best for patients if we're accessing their data and materials. And finally, our consent form is extremely explicit, and we focus on open science. I find one of the fascinating things is when we talk about open science with patients, the most common question we get is, isn't this what all science is like? So when I encounter other translational researchers who have concerns about the risk to patients with open science, I would encourage them to have that dialogue directly with their patient communities. So I would like to invite all of you to join us in the next phase of our development. As of now, we are live. Our data is being made open and available to the research community, and we're entering a phase of co-development. This is not a final release. This is a work in progress, but we actively want to hear from you to make sure that we're developing a tool that is of concrete and practical use to you in various communities. and that includes researchers, both academic and industry, patients and their disease networks, and we are actively looking for your feedback. So when you log in, you're going to be asked if we can contact you for feedback. We have an email address and a Twitter account. Please feel free to reach out to us, because this is really a remarkable accomplishment that reflects hundreds of hours of hard work from our development team. Finally, this is made possible because we're in the right environment. Without the contributions of our clinical research unit, we would never have been able to consent these thousands of patients. We are able to turn these into meaningful cell lines because of the work of Dr. Fawn and Dr. Durkin in the Experimental Drug Discovery Unit. And the LORIS infrastructure was empowered by the work of Alan Evans and his team in neuroinformatics here at the Neuro. So as of today, we have released the clinical information, including neuropsychiatric testing, genetic information, and imaging information for thousands of patients with Parkinson's disease, ALS, and neuromuscular disease. And this really reflects the hard work of a team of developers, specimen processors, policymakers, lawyers, and ethicists, and our patients as well. So I thank you for joining me here today, and I really hope to hear back from you as you interrogate our collection of patient information and data. Thank you so kindly.